thank you so much for inviting me to talk today. So I work at the university, and my talk is going to be slightly different from the other ones from this evening's talks. So I'm an academic, and I'll be talking a lot about sort of research and innovation in that space. So you may have heard of the Christabel Pankhurst Institute, and so this is a fairly new institute that's been set up, and I've proposed a new theme on multi-omics. So a lot of the talk today will be focusing on this multi-omics aspect. Okay. So just a brief introduction into the institute, if you don't know already. This is just a clip from the website, and essentially this institute's been set up to really look at health technology research and innovation, but also support that translation of research into practice. So a lot of funding has gone into this, and we have a lot of groups involved with this. So everyone from academics to healthcare professionals to businesses and local government as well. Okay. So as I mentioned before, we've got these groups that are involved, and the key thing that I've highlighted on this particular slide are the research themes. So if I can navigate this smoothly, I should be able to pull up a pointer. Excellent, right. So here's the current research theme. So we've got this digital and AI aspects, and that covers a lot of work on building sort of algorithms, working this health system aspect. And one of the things that you might be quite familiar with is connected health. So things like using wearable devices, collecting data from that, and how we can interpret that for healthcare services and healthcare purposes. And another one is also advanced materials as well. So this is a whole other dimension where healthcare can benefit from. And the theme that I'm going to talk to you a bit about is this multi-omics aspect. So a lot of you here today are interested in this whole genomics and how we can use that for a health perspective. And hopefully this new theme is going to aim to answer some more questions in that area, but actually go beyond just the normal genomics and the limitations of genomics. Okay. Right, so my first question to you guys is, what do I mean by omics? So what are omics? And I can't quite see the chat at the moment, but if you want to maybe contribute your ideas, or if you're in the audience, shout out and type in the chat. So what kind of omics can you think of? I've already mentioned one. Can you think of any other examples?
Metabol.
Oh, okay. I think I heard metabolomics. Oh, I can see proteomics. Excellent. Any others?
As well. Metabolomics.
Metabolomics. Brilliant. Yes. Yeah.
Was proteomics that?
Proteomics. Yeah, that's been said in the chat. Any other ones?
Say that again. I think we didn't quite catch that.
Sorry, proteomics has been mentioned in the chat. Are there any other takers? Transcriptomics, excellent, yeah. Metagenomics, brilliant, yes. Any other takers?
Epigenomics?
Yes, absolutely, epigenomics. Brilliant. Okay, thanks so much for your interaction there. Okay. So I'll just show you a few here now. Some of the names you've already got. So you mentioned epigenomics, proteomics, metabolomics, transcriptomics, but there are many, many more. And this is just to list a few of them. So obviously we can go into the microbiome, we can look at lipids and lipidomics. And there's interesting names as well for phenome and exposome. These are also looking at environmental factors. So we're moving just beyond looking at the molecular data, but also into this environmental aspect as well. So this is an ever-growing field, and there's many data sets out there that we could be working with and we could potentially learn a lot from. Great. So thanks so much for having a go at that exercise. And what I've just summarized here, again, is what we mean by omics. So we often call it omics or ohm, depending on the type of molecular information. But ultimately, we can use a range of technologies and analysis approaches to try and get some understanding. So genetics and genome is the same field. And for example, the microbiome is understanding all the microorganisms in a specific entity. And this is now starting to build into this next stage of multi-omics. So we're very good at analyzing each individual ohm or omics on its own, but how good are we at combining all that information together? And that's sort of the next big stage, really. And so being able to integrate and analyze this several omics data into a single analysis pipeline or within a single study is actually an area of huge interest, and lots of groups around the world are investigating that. And there are other terms to describe these several layers of omic data. And so I've just described that at the bottom in this bottom box. Multi-omics is a very commonly used term, but it can also be called integrative omics, transomics, polyomics, or even panomics or panomics, depending on which phrase you prefer. But ultimately, it all means the same, trying to analyze more than one data set or trying to utilize one or more data set. Okay. So we have to understand this process of analyzing multi-omics data. So on this slide, I've simplified that into 3 key steps. Now, this is a very big oversimplification of that. So step 1 is where we actually get hold of that data. So the biologists in the lab, so that could be any of the labs, for example, in academic labs, or it could be in companies, we can collect that data from, for example, biological sample. That might be blood samples or a tissue sample. And we can collect that data there. So that could be your genetics data, that could be your transcriptomics data, it could be metabolomics. And then we move into step 2, which is the nice bit as well. This is where we start to branch out from just the science right into other fields. So this is analyzing that data set. So we can do this as scientists, but we might actually want to do that with statisticians, or we might want to do this with data scientists. And so it's a multiple effort here to get that understanding from this data set. Once we've done that, we then get a better idea of this clinical application of what we've analyzed. So for example, if we found a particular biomarker for a disease, then we can put that towards something like a clinical trial to validate that. And so we actually fall into the stage 3, where we're trying to translate those findings into a particular technology or particular clinical application there. So I've just highlighted this again. So step 2 ultimately is a lot of interdisciplinary work, teamwork, collaborations. And then step 3, which is the ultimate aim really, is to get that healthcare application or healthcare technology from all of this. Okay, so I'm going to jump back a bit. We talked about single omics before. And one of the most obvious examples I think we've all come across already is the use of genomics in healthcare and how it's actually growing a lot at the moment. So the current omic technologies that we are using in healthcare, for example, is gene therapy. And I've just put names of Genomics England and UK Biobank as they're big names in this sort of area and working with rare diseases and detecting variants for them. So you may be familiar with a lot of that already. But also, just to sort of give you some pointers as well. I've put this link right at the bottom of the slide to also point out that the government are also working on pushing some more genomics and genetic medicine into regular, well, ultimately routine care and routine practice. So there's this really interesting link here if you want to visit the website and have a look at that. So yeah, there is a huge drive for genetics at the moment, but what about the other omics? Let me just get my pointer back. Okay. So we've kind of established that we're, as a country, we're pioneers in the genomics field, but what about this multi-omics? Why aren't we connecting other data sets? Surely we should be doing that, right? And it would make sense because if you think about the biology of this data, we've got our genetics, but we've also got these other molecules playing in our system. And so surely these should all have an impact on our health status. Okay, so this is where the challenges come in. So I've put this picture of a massive jigsaw puzzle. So combining these different omic data sets is a bit like trying to piece together a jigsaw puzzle where there are so many pieces, just like in this picture. So we end up in this sea of biological data. A lot of the data, has some relevance, has some use for us. A lot of it might not. And of the ones that we do find some use of, only one or two of those bits of information might actually be helpful for us to put into clinical application. So this whole question comes as where to start. How are we going to tackle this problem? And just to delve into some more of the challenges. So it's not just a jigsaw problem here. What I mentioned before actually is that we need a range of expertise. So we're not just talking about the biologists, we need to talk to the technologists, we need to talk to the analysts who can help us pull all this information together. And the technologies that we require can be vastly different from each other. So from sequencing, for example, or microarrays that we commonly use with genomics or for transcriptomics, we might have to delve into more of the mass spectrometry or NMR or other types of technologies that are often used in isolation by other omic fields. The third problem that I've picked up here as well is we need a large sample size. And that can be a big problem, particularly in studies where you're looking at rare diseases and when the prevalence of a particular condition is already very low. So to try and hit that target of getting statistical power with a large enough sample size can be quite tricky. But it's very important to bear that in mind when we design these types of studies. So moving on from that as well, once we've got all this data set, we need to know where to store the data. We need to know whether we're meeting the data ethics requirements and the data sharing requirements. And also that's quite important because we need to be able to share that data with other people in order to get the most out of collaboration and working on a project. And finally, but most importantly, we also need to make sure the clinical need is met. And sometimes engaging with the patient and public groups throughout a project is a good way to sort of keep in tabs with that clinical need or keeping in contact with the healthcare professionals. But that can be quite challenging in itself as well. So finding the right people to engage with, as well as doing it regularly enough, but also ensuring that you're not going to get any bias out of that as well, and ensuring that you can meet that need ultimately is a challenge. So yeah, if you can think of other challenges as well, feel free to sort of shout out or put in the chat. But yeah, these are just some of the key ones that I've pulled out for certain. Okay. So I've loosely called this the pathway to health innovation, but this again is a very oversimplified step-by-step of what you might expect. But in the Institute here, and this new theme that's been set up, and which I'll be leading, is going to focus on helping the researchers pull together their ideas and their aims in this whole theme of multi-omics. But it will also help, this theme will help us support the research groups to engage with the right stakeholders and expertise, because we have that all around us. We have all the right groups and the right knowledge, but it's just engaging with them at the right time that can sometimes be the challenge. And of course, with research, applying for funding is the biggest issue as well. So to build that sort of case, the larger the effort is, the more likely you are to succeed in that sense. So that's also a very key area that we look to support with, but also developing those innovations ultimately as well. So we've got this pathway where we've gone from research. If I get my pointer back on. So we're going to go from this point of research and collaborations, which is where pretty much a lot of research groups are in the multi-omics area. But to move into this area of product or service development and then commercializing and then getting into healthcare setting is actually the toughest part. So actually to try and bridge those gaps, the Institute is aiming to sort of pull together this community and help get the right people on board at the right stages. So once we've sort of met the challenges of the research side, we can then start thinking about these latter stages as well. And that these latter stages have worked very nicely with some other research areas and technologies, which has already shown that this can potentially work. Okay, so here's the most interesting part of this theme, is what exactly can we do with all this multi-omic information. So I've just put up 4 rough areas of where I think some of the key areas of innovation will be. So the most obvious ones would be building a diagnostic tool. So I mentioned rare diseases a few times, and being able to diagnose some diseases is actually quite difficult at the moment. And sometimes we find that when we've done research in a single omics area, we find that it's actually not just that single omics on its own in that isolated area. It's actually multiple things that are happening. And this is where a pathway can be involved, a biological pathway that can then lead to a particular disease. So having a sort of diagnostic tool that will screen for certain parts of that biological pathway is actually quite important. So for rare diseases, that would be particularly useful. The next stage, idea I've mentioned here is just about medications as well. So this whole idea around safety and drug interactions, we can learn a lot more in that area if we look at different types of omic data. Another area that a lot of people have been working on already is to try and understand complex diseases. But the next stage from understanding that is to then actually prepare some, a technology that can help improve treatment plans. And that's the stage that is most relevant for a clinical setting. And so this is an area that is quite untouched at the moment because of all the challenges. Many research groups haven't actually managed to get to that stage yet. So obviously we can see that pathway a bit better with genomics, for example, but to see that with multi-omics would be the next big thing, really. And then finally, in the way that the government is also trying to push having more personalized medicine with genomics, trying to get multi-omic profiles into routine healthcare or into personalized medicine, in essence, through the system, for example, of electronic health records, could be another area of where we could do some more innovation. Okay, so that was a sort of quick whistle-stop tour from me, and I've just left this last slide with some contacts. So at the moment, this theme is quite new, and I'm building together a lot of researchers who are interested in this area. If you're interested in this specific theme, then please do contact me. So this first email address is mine. And feel free to drop me an email, and I'd love to talk to you and take this conversation forward. If you're interested in some of the other themes that I mentioned from the Institute as well, do drop the Institute a message at this second email address. Or you can also sign up to their newsletter and keep up with the updates. And you'll also get updates from this multi-omics theme through that newsletter as well. Okay, so that is it from me.